Well, welcome everyone. My name is Dr. Liz Pierce, and I am the moderator for this series. And I want to welcome everyone to our first session. We have today Dr. David Ussery, and he grew up in Springdale, Arkansas. He got his PhD in biochemistry from the University of Cincinnati College of Medicine and did a postdoc at Oxford University. He has lived in Denmark for 15 years before returning to Arkansas, where he is the Helen Adams and the Arkansas Research Alliance Chair in Biomedical Informatics at the University of Arkansas for Biomedical Sciences. He has worked with genomics for more than 20 years and is currently working on several projects related to microbial genomics, including SARS-CoV-2. His work combines high throughput experimental and computational methods in order to aid in rapid diagnostics. He is currently co-lead in the data lifecycle and curation part of the NSF EPSCoR Track 1 grant for the state of Arkansas and director of the ARC-GEM Center. And today he's going to talk about some of his recent work on comparative genomics of SARS-CoV-2. There are now close to 100,000 corona genomes available, with the number growing daily. For faculty and students working on big biodata research projects, data curation is an essential part of working with big data, especially messy biological data. And with that, I'm going to turn it over to Dr. Ussery.
Okay, thank you. Can you guys see my screen?
And yes, everything is looking good.
Okay, and Liz, now can you see? Is it okay? Is it big screen now, full screen?
It looks great.
Okay. So, I'm gonna just a second. I'm gonna get a little pointer so that I can, let's see here if this works. And then I type play, yes, and then I do this. This is actually, I'm going to go back. There. Okay. So you can see this red dot. Can you guys see this moving around?
Oh, yes, we can.
Okay, so I want to point out, so this is kind of my team. And you see there's a lot of diversity here. So this is Skylar. Skylar just got her PhD this summer, a couple weeks ago, and she's from the UALR, UAMS Bioinformatics program. So Skylar is one of the students there. There's also Kanishka, who's another student in the UALR program, and Daniel is also. So we have several students there. We also have students part of the GPIMS program here. This is Dua. She's hopefully going to graduate this autumn around Christmas time, and so is Visanu here. So you can see we have a lot of different people here. This is Seiran Jun, who's an assistant professor in a group, and Mike Robeson. Intawat is an associate professor. Back hiding behind here is Caleb, and this is Parun and Tip, and they're responsible for a lot of sequencing. So anyway, it's kind of a big group. But this is, like Liz said, the first seminar in this Integrating Bioinformatics Education series. So now, you guys have already seen this. Now we'll transition. And for those of you who've seen me give talks before, you know, the second talk is always a picture. This is from Rattlesnake Ridge, the park close to here. Okay. And you've seen this. So this is basically the first one. And I think, Liz, they're every Friday now? From now on for the rest of the semester, something like that?
Yes, thank you. And I appreciate you bringing up our webpage. For anyone who is interested, we have a 12-week series, and you're welcome to go to our webpage, ualr.edu/bioinformatics. And to check out our Integrating Bioinformatics Education series.
Okay, good. So this is the first talk of 12, and there's a little what basically Liz had read earlier. And so here's the outline. Now, like a lot of people, things are very busy. So I'm going to talk about basically three things: Arkansas and COVID-19, kind of a little bit of context. Then I'm going to talk about SARS-CoV genomic epidemiology. There's actually a book out on this, believe it or not. A fairly thick book, like 250 pages on genomic epidemiology of SARS. And I'm going to talk a little bit general about data curation. But having said that, that was my original plan. But as things begin to rush, I also had to teach a class today, and I'm working on a grant on, you'll see later. But so I wound up just, we'll see. So the first thing about the background in Arkansas, and this is a figure in a grant that we're writing, an NIH grant on looking at COVID-19 wastewater. So this came out in April. What they did is they looked at the 50 states and said which states are going to be especially vulnerable. And Arkansas wound up number 3 on the list, which was kind of surprising in a way. But on the other hand, the way they define vulnerability, they're looking at the diabetes in the population, obesity, health risk problems. And I love this quote at the end, because for those of you that have been in, I grew up in Arkansas, we used to say, thank God for Mississippi, because Mississippi is the last and Arkansas is next to the last. And when I was growing up in the 1960s, 1970s, when Bill Clinton was governor, he used to say, thank God for Mississippi, because they were number 50 in education. And so we weren't the worst, we were second from the worst. And so several things make people from Arkansas at risk for COVID-19 in terms of like the age of the population, but also other things. Like I said, diabetes is a big risk and other things. So well, anyway, that's just worth kind of talking about. So the next slide is kind of this is now September, but basically earlier this summer I gave a talk, and what I did is basically just say, okay, in one day, this day was the 24th of July, what's going on with COVID-19? And on that day, 1,150 people died in the US. 74,000 people tested positive. In Arkansas, 900 people. And if you guys read the newspaper today, do you remember the news when I was reading it this morning? Another 969 people tested positive today, or well, they announced it today. It would have been yesterday for COVID-19. So this is not really going away. So the other interesting thing is on one day, 1,869 new genomes for SARS has been submitted to this database. Just one day. They're averaging between 1,000 and 2,000 genomes every day being deposited. And the last thing, which is, from a scientist's point of view, is pretty frustrating. It's good news, bad news. There are 341 scientific papers published on COVID-19 in one day. And if you think about that, just to read one paper takes me a couple of hours to try and read and understand it. I couldn't read 340 papers in one day. And even if I could, by the time I'm finished, there's another 300 papers the next day. So this is a daunting task to try and keep up with what's going on. So here we have the SARS virus. And it's basically an RNA with a coat around it, a lipid coat, and it's got these spike proteins. And as probably you all know now, the spike proteins binds to a receptor called ACE2 for angiotensin converting enzyme. There's more than one, so it's number 2. And that's how it gets into the cell. Okay, so we will talk more about that in a bit, but now I kind of come to my disclaimer part for this talk. I usually always, fairly early in the talk, kind of give a disclaimer. And this time around, the disclaimer is that, like I said earlier, I was running out of time. What I've done is, about 2 or 3 weeks ago, I gave a talk for students there. We're going to do rotation projects as part of one of the graduate programs at UAMS. And these are 5-week rotation projects. So basically, these are projects that we could do, just something to kind of get a flavor for what's going on and to try and recruit students. And I did manage to get one student from the pathology department, but he's doing a project with me, but it's not with COVID-19. But anyway, and that figure that I gave Liz came from that because I had this. So this is the figure here, and I'll spend a bit of time on it. And this is basically part of a National Science Foundation grant that we got funded this summer, and the project is called DART. And basically, it's a big data proposal. It's about big data infrastructure for the state of Arkansas. And actually, the nice thing about it is a fair amount of money. As you can see, $24 million over 5 years. This started in July. And so John Talbert there at UALR and myself are leading one of these parts that's on data curation. And so this is part of this. It's called data lifecycle curation. And the whole grant is very, very hardcore computational. And this is called DC for data curation 3. It's the third part in this proposal. And it's a lot more biology. And so what John Talbert's idea is that one of the rate limiting steps with big data is that often you have noisy data and you spend a lot of time cleaning it up. And so if you could automate cleaning the data, this could really speed things up. And the idea, the analogy is like you have a washing machine and you have all your data, you have all this data in there, and you throw the data in this washing machine and out pops clean data that's curated. And so in our case, and again, this is about a month ago, we had 70,000 SARS coronavirus 2 genomes available for this website. 70,000 genomes. And you've probably seen stuff in the newspaper sometimes. Oh, it's mutating fast and all kinds of things are happening. Well, it turns out there's varying quality. And some of them are really poor quality and some of them are good quality. And so you say, well, how do we know what's a good quality and not? Well, we know the genome should be about 30,000 nucleotides. And this is, they sequence the first one, good quality. So we could look. So we say, is it the right size? That's a fairly simple question. And you could score it. If it looks about 30,000, in that range, fine. But if it's less than that, if it's only 25,000 or 20,000, you start getting dinged. You lose points every time. We also look, are there 4 bases GATC? If there are, fine. If there's bases other than that, you start losing points. Is it in one piece for the whole genome? Yes, okay, fine. If not. And so what we do is we basically can take all these, there's a lot of different things we could do, and we get a quality score. And you can do this automatically. And so what we did is we started with 70,000 genomes, and we put it in our washing machine, on our data curation, and we looked at the quality scores. We said, we want anything of a quality score of 0.9. And that left us with 17,000 genomes. So you can see that but these are clean now. So these data in a sense has been washed. And we can use these, we can build trees. And there are maybe 5 or 6 known kind of clusters or clades. They're called phylogroups. And if you see here, when the outbreak first started, there's one genome sequence. And then it began to change just a little bit. There are kind of 2 clades, and there's 3. And then these begin to split some more. And so now you've got 5 or 6 major clades. But these all differ just by a few nucleotides. And so when people come in to be tested, you take it like a nasal sample or something and you're tested, and there's a little bit of liquid left over. And you can take that liquid that's left over, and there's still virus in there. And the ones that test positive, we can look for ones, and we picked 3 UAMS patients that tested positive that were quite high viral loads. And then what we could do is sequence the genome. And what we found is that these 3 patients, they have genomes through 3 different clades. Now, as we sequence more, we'll probably find with this and some with that. But what this is, if these 3 people were infected from different viruses, and these reflect, this clade might have come from Europe, this clade might have come from China, this clade, one clade could come from South America. So you can kind of get an idea. And even in the US, there's kind of an East Coast versus West Coast. There's a lot of coronavirus in New York. There's a lot of coronavirus in Washington State. And so we could use this for what's called genomic epidemiology, for kind of tracing where it came from. But this is all great, but you can only do that. You really need to have good quality genomes from that. This is part of the data curation program. And one of the things that we could do is we could take some of this data and we could actually look at conservation. So here is the coronavirus. This is the first genome that was sequenced in late 2019. It's 29,903 base pairs. And so this sequence, we put it as an arc. It starts here, and that's 1,000, 2,000, 3,000, 5,000 bases, nucleotides, all the way around. And what we could do is we look at the variation within the SARS. And basically, we're looking at, this is interesting, okay, I don't know how many genomes we did here, but the red line here is looking at variation amongst the different strains. And you can see that there's some regions here where they're variable. And these regions, here's the N nucleocapsid protein at the end. And these regions that vary are things that you could use for looking at, well, if you know this variability, you can use it in potential in tracking and diagnostics and other things and keeping track. And so anyway, so that's just a way of looking at the whole sequence. And this is an RNA genome. And you can sequence this either directly sequence it, you can make an RNA copy, a cDNA, an RNA copy of the DNA copy of the RNA, or you can sequence the RNA directly using these, what's called third generation sequencing. And so we've been playing around trying to use this on isolates from UAMS stations. So that's kind of the first project. The second project is looking at the spike protein binds to this receptor that's called ACE2. And the ACE2, it actually does things normally in the cell. And if you look at where the ACE2 lives, mainly lives in the small intestines, and some in the testes and in the kidneys. And you see it some in the lungs, but this has to do with the relationship between the intestine and the lungs. There's this kind of lung microbiome axis between the gut microbiome. And so it shouldn't be surprised because of this. Here's where most of these ACE2 lives. So it should be surprised that when you have an infection, a lot of it could be through the intestine. And so that's something to take into account. And it does infect the lungs as well. It causes a lot of other problems, but the main receptors are mainly expressed in the intestine. So normally, this ACE2 binds angiotensin 2, which is a small, short peptide that's only 12 amino acids long. And it converts angiotensin 2 to angiotensin to a different form where it just cleaves off the two amino acids. And so this has a lot of downstream effects. Basically, the ACE2 here, when it cleaves angiotensin 2, it can actually reduce a lot of inflammatory response. So if you're injured, you have angiotensin 2 being presented, and that's kind of a sign, it's a signal that something bad happened. So then ACE2 cleaves this and can kind of calm things down. If you don't, if ACE2 is binding now, see here's with the coronavirus here, this is messing it up, so it can't bind to angiotensin, and now angiotensin 2 builds up and causes a lot of other problems, and eventually you're going to get inflammatory cytokines. And in some cases, you get really, really bad cytokine storms. So what ACE2 is normally doing, ACE2, so here is what's called, this is the precursor protein here. This is a big protein that gets cleaved in the kidney by renin, and it cleaves this end off right here. And then the first part of this is a secretion system, and that gets, when it gets secreted, you wind up with this short peptide that's only 10 amino acids long. And here's the sequence for it. And then ACE1, angiotensin converting enzyme, will cleave off this leucine at the end. And so it goes from 10 to 9 amino acids. And then the ACE2 will cleave off this phenylalanine and histidine here, these last 2 amino acids, and it's just got a proline there. And this is an inactive form. And so this form is basically everything's safe and you're okay. But this form, actually even this form, can be, this is angiotensin 2, this form can be a signal that there's been damage, and when it builds up, you're going to get the inflammatory response. So ACE2 is a really good guy in preventing a lot of cardiovascular problems. You can kind of think of it like a bit of a teeter-totter here, kind of a balance, where if you're injured, you have this angiotensin 1 building up, angiotensin here or angiotensin 2. If the ACE2 is active, it can convert it to this smaller form, and now this is protective. So this is injury, this is protective. So ACE2, you want to get to this state. And so this is an important thing to remember. One of the strategies, sometimes I read in the paper, people talk about down-regulating ACE2. But if you do that, you're going to mess up your cardiovascular health. So that's probably not a good thing that you want to do. So, and again, so one of the things we could do is we're trying to look, ACE2 binds not only angiotensin, but it also binds bradykinin and several other short peptides. And in general, it's going to be cleaving. You have a proline followed by an aromatic group, and you usually have an aromatic group over here too. In this case, there's a tyrosine. And so what we can do is build up models for this, where you can see this is the proline, this is the histidine, isoleucine, tyrosine, valine. So you can build up models for this, and you can look across. In this case, we looked across 113,000 human proteins. So there's around 20,000 genes in the human genome. And those 20,000 genes make 100,000 proteins, because you can have alternative splicing, a lot of things, like I showed you earlier for angiotensin, it gets cleaved and other things. And we found 58 matches out of this, so we're kind of looking to see what some of these do. But the idea behind this is that ACE2 can basically offer protection. So anti-hypertrophic, anti-fibrotic, vasodilation, antioxidation. So these are all good things. Here, these are bad things. And so the presence of ACE2 is actually very important for cardiovascular health. This is the bottom line. That's kind of where we're going with that. So that was the second project. Sorry. So the third project is looking at a high density peptide array. And so this is, here's your SARS genome. So this is the virus, and inside is the RNA copy. It's a messenger RNA, single-stranded RNA copy, and it's surrounded by lipid coat, and it's got this spike protein. Remember, the spike protein is what binds to the ACE2. And so if we're to take this RNA genome and spread it out here, you have two large open reading frames, which makes one big polypeptide up to here. And this gets cleaved into a whole bunch of smaller proteins. And then you've got all these other products here. These all have poly A tails. These are all polyadenylated. And these are a lot of the proteins that are responsible for the activity. Now, this is one big message. And so one of the things you can do is one of these proteins in here, in the I think it's ORF1A, that gets cleaved, one of the small proteins, actually interacts with the spliceosome, and it affects alternative splicing. So here's an example of a splice here. You've got a 5' 3' break point, and you can splice things together. And so you can see here where you can splice. This is actually this genome gets spliced, where part of the beginning gets spliced into the S. And you can have all kinds of different things. So you wind up with different transcripts. And you can see here's copy numbers. And so see here you have all these. Now it turns out that the N protein at the very end, this is the nucleocapsid protein. And this is a protein that's responsible for binding with the RNA. So you have lots and lots of that. And so you have lots of this. This is at the very kind of towards the end of the long polypeptide. This is high copy number here. And so you're getting varying amounts of these, but now with alternative splicing, you're getting different products in different portions. So this kind of makes it a little bit complicated, and so you can look at different splicing junctions, but also in the host, you have alternative splicing as well. So one of the things we want to look at is taking these peptides. We can take a short peptide that's 16 amino acids, and we can put it, basically, you think about like a glass slide, you could attach it. And we have these high density arrays where you can fit 4 million of these 4 million different peptides. And so we look at unique 16-mers. So my friend was talking about doing this, and he wanted me to help him calculate all the possible 16-mers. So we went through all the coronaviruses. So there's more than, at that time, I think there was 50 or 60,000 SARS-CoV-2 genomes, but there's a lot of other coronavirus genomes that are in the GenBank. So what we did is we looked at everything we could find, and we went through all the proteins that we could find, and we wound up with 1.9 million proteins, or 1.9 million 16-mers, unique 16-mers. We could fit 4 million on. So what we decided to do is look at these modifications. So we look at editing, alternative splicing, and frame shifts. And so if we do that, we find another 3 million proteins. And actually, so we have this 4 million peptide array that we can use. And what we could do is this is every possible 16-mer that you could get. And then what we're going to do is you can take antibodies. You take a blood sample from a patient that has COVID-19, and you see which of these peptides do the antibodies bind, so you can realize what's being raised against it. And so this is, so basically you can print these things, and this is a real simple microarray. So here you've got you're binding your immunoglobulin, and you're looking at fluorescence. And so you've got these little peptides on there, and you can see what's binding, and you get colorimetric changes. So this particular array is looking at 67 antigens. And so you can see where the antibodies are binding. Now, here's common cold. 4 of the strains of coronavirus actually cause common colds, or RNA. And you can see antibodies lining up for that for different people. And then not for this would be SARS-2, 2019, the new coronavirus. This is the MERS, and this is SARS. And so what you could do is you could look across. And one of the things we were wanting to do is figure out which particular proteins are there. So what we want to do is we want to be able to test 1,000 COVID-19s. And this is with the collaborator in Washington State. We can also do this in Arkansas. And as control, we're going to use 2-year-old frozen blood serum. So this is basically patients before. So those should test negative here. But you will see lots of things from the common cold, from coronavirus that cause a common cold. And so that's kind of going to be some fun things to look to see what's there. Okay, the last project is kind of fun. This is looking at epidemiology. And so this is, 1854, John Snow. So these are different, the X marks where different wells are. And he found that looking at looking at Vibrio cholera, people that were infected around this particular pump. And what he did is he locked this pump, and then the number of people infected with cholera went down. And so the idea is that we've got collaborators in Conway Corp, the wastewater treatment plant there, that are letting us test some of the water. And we can do RT-PCR. We sent it to some collaborators in Washington State. And eventually we're also doing this in Northwest Arkansas, at Fort Smith, in Greenland. I don't know if you guys know where Greenland is, but it's just south of Fayetteville. And Fayetteville, in Springdale, in Rogers, in Bentonville. And we also have in Conway, and in Little Rock, and in Pine Bluff. So we're looking across. And the idea is to be able to do this. And so this is part of the grant that we're applying for down here. A $4 million. It's $2 million a year. It's called a UO1. It's a project grant, and it's due 15th of September. And actually, it's due a week from today in terms of the internal deadlines for UAMS. And so that's why I'm a bit scatterbrained and going crazy. We're having a lot of fun with that. So this is just showing that this is from a paper where you could show that if you monitor in the wastewater versus hospital admission, you can basically see as you see more COVID-19 in the wastewater, a few days later you see more hospital admissions. And here you can actually look at how much virus you get per mil at hospital admissions, and you can see a pretty good fit here. And so the bottom line is it looks like this is probably going to work, and it's kind of so this is from in the Netherlands where they're monitoring February 27th. There's nothing there. February or March the 5th, you begin to see things, and March 16th, you begin to see higher levels. So Schiphol, this is the airport there. So, and I don't have a fancy ending. That's more or less it. It's a short talk today. So Liz, I'm not sure how long my talk was supposed to be, but I usually figure it could be more of a discussion. So I'm done. For questions. Any questions?
Yes. Well, thank you very much. Let's open it up for questions. And I invite folks to please, I'm monitoring the chat. Please post your questions so Dr. Ussery has a chance to respond. So we'll take a moment to let folks collect their thoughts and to post a question.
I see Segam's on there. Hi, Segam. I'm looking to see if I recognize any of the people on the list. And Mary Yang's on there, and John Talbert. Anybody have any questions?
Oh, Imran was
How do you
curious.
do automated cleaning?
Yep.
Yes. So the automated cleaning, and I kind of talked a little bit about it, but I can go into a bit more detail. That in this case, for the viruses are literally a million times smaller than the human genome. Viruses, this is only 30,000 characters long. So you have them, and you know what it should be. It should be 30,000, 29,000 nucleotides long. So what we do is for each virus, we ask some simple questions. Each sequence, is it about the right size? Is it 30,000 or 29,000? And we say if it's between 28,000 and 30,000, okay, fine. If it's less than that, we dock it some points. We ask another question. We say, are there 4 bases? Is it all GATC? Are there bases? Sometimes if you don't know what it is, you put N, okay? Then it gets docked for that. And so we have a bunch of different criteria, and we use that criteria to set up what's called a quality score. So for each of those 70,000 genomes, we have a number in the end, and then we can filter by that number. And so the automated cleaning is we just say anything better than this. Does that answer your question?
Dr. Ussery, the next question is from Syed Jamal. He wants to know, how do you test the difference in antigenicity of different clades? Are these clades in different parts of the world?
So that's an interesting question. Yeah. So we don't know, but most of these clades are only different by 1 or 2 nucleotides, and some of the clades will virtually make the same proteins. So some of the mutations are synonymous, or they're not going to really affect. So I mean, I don't think in terms of antigenicity they're going to have a big effect. They might, but well, that's something that we could test because we now have sequence from lots and lots of patients, and then we can take some of the blood from the ones that we know this type and blood from those that type, and see which antibodies are binding, where they are. So we could do that. That's easy enough to do experimentally. So I see Michael Brewer ask a question. In the data washing machine, were there any interesting correlations between the data that got cleaned versus the data that got used? Do you feel there's meaningful data loss? Well, so in our case, this is just kind of a really simple cleaning method. We say, by good quality, we mean, is it the right length? Does it have all G's, A's, T's, and C's? Is it in one piece? Is it in many pieces? And so from my perspective, we're throwing out the bad quality data. There might be something useful in it, but yeah, I don't know if that makes sense. But did that answer your question, Michael Brewer? Okay. The sensitivity of microarray data. So the sensitivity, so it's actually, this is an ELISA kind of assay, and so it's fairly sensitive, I think, and you're looking at fluorescence of binding the antibody. And what we're doing in parallel is we're making predictions. So some epitopes we think would be very strong binders, some would be weak binders, and nearly all of them are not going to bind at all. So out of our 4 million peptides, probably 3,900,000 aren't going to bind, but there's going to be few that are. And we've made some predictions, and we're going to see how good those predictions are. And we did this for Ebola, and we could actually predict from the Ebola sequence what were likely good epitopes, and it actually worked fairly well. It'll be interesting to see whether it works for this or not. So let's see.
Thank you.
And Purvi had a question. She wanted to know, how do you plan to use or build truth data set comparisons? How metadata acknowledgement being used?
So I'm looking at the question, any improvement down the line for data quality. So yeah, and there is metadata as well that obviously we can incorporate, and we can look at geographical background and where are they physically, where was this sample collected, other things. There's a lot more that could be added that would make the data more useful. And with data quality as well, as some of the sequencing technology gets better, we can begin to get a better idea, because with sequencing, you could get along with the sequence, you could also get another file that's basically looking at the quality of each read as you go along. And so if you had that, you could add. So there's a lot of things you could do to improve the quality, I think. We're not done yet. So.
Dr.
I see that
Ussery?
question now. How do you build truth data sets? Yes, yes.
And Dr. Ussery, for students who are interested in trying to do maybe some of their own studies, do you have any recommendations on data collections or data tools that students and faculty might want to take a look at?
Well, so, and as part of this EPSCoR, as you know, we have this Embry workshop in the spring every year, right? Hopefully we'll have it again this year. And that's a good place to start in terms of there's our biotools. And now as part of this EPSCoR, what we're trying to do is reach out to colleges and universities across the state of Arkansas for undergraduates and even high school students. We're going to try and have several one-week workshops where we will provide some of the tools and give an introduction to some of this.
Yes, and for folks who are listening, we are hoping again in the spring that we can offer our Genomics Workshop, which is really open to undergraduates, graduates, and faculty who would like to have a terrific, I'll put in a plug here, it's a 2.5-day workshop where you start actually going from a DNA sample, and you take it all the way through sequencing analysis to answering a number of questions about that sequence and presenting your results.
Yeah, and I'm hoping we'll see. I'm hoping we can maybe make it even longer this next time, maybe 3 or 4 days. But the bottom line is on Monday morning, the students pipette a sample. We give them a sample with some DNA in it, and they pipette it on, and then by the first day they have enough reads they can begin to figure out what it is, and by the second day they have the whole genome, and then they spend the next day working on that, and then the third day they do a presentation about what we gave them to sequence. So it's a combination of kind of experimental and computational.
And I'm just curious, in terms of future projects, Dr. Ussery, is there a particular question about SARS-CoV-2 that you would love to be able to unravel? What do you think are some of the biggest mysteries about this virus?
Well, there's two things that I think that are really important. One is with the virus itself. We would like to sequence the RNA directly, and there's been a couple people that have done this. It's a little bit tricky, and you need the people that have done it. You basically have to have a special lab, a biosafety level 4, like a germ warfare type lab, and you need lots and lots of virus, and it's just not practical. But with the technology, as it's improving, in principle, it might be possible to get it so that you could do it in a normal lab, like here at UAMS. And to sequence the RNA directly, the virus, to sequence that, you could get a lot of useful information, because in the RNA, some of the bases are modified, and this controls virulence. So you could have two different strains of that would give you the same sequence when you're to sequence it using RT-PCR, but they have different methylation or different base modifications, and that can have sometimes very big differences in how infected the strains are, and a lot of phenotypic differences. So one of the things I'm very interested in is that kind of thing, how the base modifications of the RNA could affect. The other thing that I'm really interested in is better understanding the host physiology in terms of this, for example, this ACE2, this protease that's cleaving other things. And it turns out we've known for 20 years now that ACE2 cleaves about 7 different proteins, but it probably cleaves more. But no, it's been really kind of difficult to do a systematic study of this. And, before early this year, before COVID-2 came out, not that many people were interested in it. But now all of a sudden, of course, it's a major research area. And I think now that we're realizing that there's a lot of regulation that's going on at the level of these small peptides that are single molecules. So I'm very interested in that. And then I guess the third thing, I said there were two. Of the two, the third thing is I'm very interested in what would be good epitopes for this. And if you know a good epitope, then you can figure out how to build a good vaccine, right? Based on something that would give a very, very strong immune response. So that's kind of some of the things that I'm interested in.
Well, thank you very much. Are there any other final questions that folks would like to ask? Oh, we had I don't know if you saw, Dr. Ussery, a comment from JM.
Yeah, sorry, I don't know. This is Jason. I don't know why my name came across like that. But yeah, that was the thing, Dr. Ussery, that my wife and our son received gene therapy, so it's Hizentra every other week, and it's more of a gee whiz for my wife and I. We're curious to see, like, over time, does he start to develop antibodies for COVID through because of the gene therapy.
I don't know enough about what kind of gene therapy he's getting. It's gene therapy for what?
It's gene therapy for his immune system. So he has.
So he has a weakened immune system. So that.
Yes, he has CMT4J is the disease that he has.
Okay.
Yeah, and his I forgot what they call it, the IG trough. It's very low, and so what it was is to bring that back up, and then he actually had it. And like I said, forgive me, I'm not a medical person, but it it was elevated and then brought to close to what we have. And so my understanding is Hizentra. It's like everyone that donates blood, there's these thousands of he has these thousands of antibodies that are delivered within this Hizentra.
Okay.
And so, yeah, and so that's like. I'm sorry. Go ahead.
Well, the question is because normally the antibodies are made for normal people are part of the immune response, and they're made from the host cell, right? And so it's an interesting question. If you're given antibodies, you can still fight it off, but I don't know how long eventually those antibodies would circulate. Eventually they would die off or whatever, right? And so I don't know whether you could get good response or not. That's so because there is sometimes what happens is if you get an infection, you have a response, right? But then you have these memory cells that remember what that is. They're very, very low levels of expression, but if it comes again, then it triggers it. So it might be in this case that I don't know whether you'd have memory or not, to be honest. Sorry, that didn't really answer your question.
Oh no,
it's a complicated system.
It is. It's fantastic. Thank you. I appreciate it. It's just one of the things that we wonder because with these thousands of antibodies that the little guy has, it's like eventually is one going to appear for COVID, and that's one of the questions that we had too, like as far as like an intervention for COVID. Could it be gene therapy? Is that a possible treatment? We don't know. Like I said, but it's more of a gene.
Well, they talked about it. I don't know if you've read about this. So they can take basically the blood plasma from people that have had a healthy response. So you've had COVID-19, you have immune cells circulating. Those people could donate blood to others and help. In principle, that blood should give you protection. In principle. So and that's I don't know if you've read. That's been in the news recently about that. Yeah.
Interesting.
Okay. Yeah. So I'm reading now. Kanishka's question says, "Is there reduced pathogenicity?" Yeah, I mean, if there is reduced pathogenicity for adapted to humans, so apparently SARS-CoV-2 is very adapted for living in bats. And the bats have a particular system where they can just kind of ignore it and it doesn't cause problems. But obviously, that's not what's happening in us. I don't know. I mean, to be honest, what would be the molecular basis? Well, we know where it binds, and we know what binds. It's the spike proteins binding to ACE2. The problem is ACE2 is pretty important. You don't really want to get rid of it. So anyway. Oh, so I see another question by Syed that says, how similar are the 16-mer peptides to Bgg Bcg? So some of them are 100% identical because there is some overlap in some of these proteins. Yeah. Phil Williams asks, are there any non-coding RNA in the genome? Yes, there's some. It's all basically, there is a messenger RNA that gets transcribed as gets translated in one big polypeptide. But there's also some parts in it that make non-coding RNAs that are stable. So the answer is yes, there are some non-coding RNAs in the genome.